Hi everyone, and welcome to our video on the applications of gene sequencing. This is part of our module 6 part 3 video series all about manipulating genomes, and in this video we're going to be having a look at specification references 6.1.3b part 1, how gene sequencing has allowed for genome-wide comparisons between individuals and between species, 6.1.3b part 2, how gene sequencing has allowed for the sequences of amino acids and polypeptides to be predicted, and 6.1.3b part 3, how gene sequencing has allowed for the development of synthetic biology. First thing then, let's start off with these genome-wide comparisons. Now, since we've actually developed techniques that allow us to sequence entire genomes, then obviously what we can do is have a look at the entire DNA sequence of an organism. Now, that's not just the bit we find inside a nucleus, should a nucleus be present, but it also looks at some of those other structures. So of course the chromosomes are one bit, but there's also mitochondria and chloroplasts that have their own DNA. And what we actually are able to do is, once we've gone through this process of sequencing these genomes, we can then store those in something we term a gene bank, which is basically like a massive data storage of all of the sequences of these different genomes of different organisms that can be accessed internationally. So this whole genome sequencing just means that we can actually go through not only the chromosomes, but also the mitochondrial DNA and the DNA within the chloroplasts. What we're then able to do is actually compare the similarities in the different species. So once we've sequenced the genomes of a couple of species, we can then put those in side-by-side comparison, and what we're going to be able to see is how similar they are, and particularly which genes show a lot of similarity with one another between a range of species. And what we're going to find is that those genes that are conserved in the process of evolution are going to be very similar amongst a whole range of different species. What we may find is that some genes have had these very slight changes that mean they now perform a new task. And perhaps one of the best examples of this that you may have met back at GCSE is the FOXP2 gene. Now, FOXP2 codes for a transcription factor that actually controls activity of hundreds of different genes. And one of the key things it's responsible for in humans is the development of speech and language. Now, FOXP2 isn't unique to humans. There are versions of the FOXP2 gene in mice and chimpanzees. But obviously, mice and chimpanzees are not sitting there chattering away like we do. So what we can do is have a look at the sequence in that FOXP2 gene and then compare it between those different species, and that gives us an idea about those subtle differences leading to what may be interpreted as quite significant differences in the outcome. So we've got this speech and language ability, whereas the mice and chimpanzees don't have that same level. In terms of what causes these differences between the different organisms, then those shared genes may have been altered to work in slightly different ways. And FOXP2 is a great example of this, because we obviously have the wide range of speech that we do, whereas mice don't. We may also see changes to the regulatory regions of the DNA, and as a result of those regulatory region changes, we might see increased protein expression, because if obviously we're altering the regulatory region, we're going to change how much of that particular gene is expressed, and therefore the amount of protein that we will generate. And one of the key things that we are now able to do as a result of all of these different organisms of which we've sequenced the genomes for, then we're able to compare their different genomes, and then we can have a look to see just how similar they are to one another. And this lets us actually work out evolutionary relationships. So what we once thought of as being closely related species, run their actual genomes, compare the differences between them. If they're very similar, they're going to be closely related and probably share an evolutionary ancestor. If they're not, probably not, more distant relation, or maybe no relation. So what we can actually do is make the statement that the more similarities there are in the genome, the more closely related those species are. The other advantage that we've got through this gene sequencing is that it allows us to actually have a look at extinct species and work out where they fall in the evolutionary tree. Because we will be able to get DNA samples from bones or teeth that still exist today, and running that through PCR prior to sequencing means that we'll have sufficient DNA to carry out the sequencing process, and then we can compare those genomes with the ones that we have today in our lovely gene bank and work out where this extinct species actually fits in. What organisms alive today are closely related to this now extinct one? The next aspect we can have a look at is the variation between individuals. Now, we do have variation within a species. If you just think about other humans that surround you in life, then you're not all identical. You show variation. And it's quite interesting to know that if we think about humans, in the species of human beings, Homo sapiens, we actually only have 0.1% variation in our genome. And that's tiny. If you think about just how different you are from other people in your class, assuming you've not got an identical twin in the class, of course, then all of those differences that you can see, and obviously ones that you can't see, are down to just 0.1% of the genome showing that variation. And the result of random mutation is causing those variations. So you've all got the same genes, but obviously we've had these random mutations that have led to variations in the alleles that we have. Now, one of the things that we can look at are these things called single nucleotide polymorphisms. And these are basically the locations on the DNA where the substitutions are occurring. And the key thing here is that there's not one hard and fast rule about what happens if this substitution takes place. We may see no effect, we may see an alteration to a protein, or it may alter the expression of the gene, because it all depends on which single nucleotide polymorphism has actually taken place. One of the kind of newer areas, I suppose, of genetics, which is actually really fascinating, and I highly recommend you read into this a bit more, is the field of epigenetics. So this is basically looking to see how your behaviours and your environment can actually alter the way in which your genes work. And a lot of this has come out of the identical twin studies they do up at St Thomas's Hospital in London, where obviously identical twins are born with identical genomes, but due to the different things that they experience throughout their lives, the different environments they find themselves in, maybe the different things that they eat or put into their bodies, etc., then they're able to identify the changes that have occurred to the genes that are switched on and switched off, just as a result of the things that you encounter in your everyday life. And obviously this is something that's highly interesting, because if we're able to turn different genes on and off, then we might be able to harness that in different medical treatments in the future. In terms of the different types of epigenetics that we have identified so far, there can be methylation of a chemical group in the DNA. And if we're methylating a chemical group, then we're actually going to end up blocking the transcription factor from binding to the DNA in that region. So typically, methylation is switching genes off, because if a transcription factor can't bind, we're not going to be able to express that gene. If we want to turn a gene on, that would be a process of demethylation, removing that methyl group. Second one is the histone modifications. Hopefully we remember that DNA is tightly wrapped around these proteins called histones, and when they're wrapped around the histones, we can't read that DNA. Those genes are going to be switched off. If we've got genes that are not wrapped around histones, they're the ones that are switched on. So what we can actually see happening is by adding or removing chemical groups to the histones, That's going to alter whether a gene is wrapped or unwrapped, therefore whether it's on or it's off. The third one is the non-coding RNA. Now, this non-coding RNA is all to do with helping control the gene expression by attaching to the coding RNA along with certain proteins to break it down. So what we do have here are 3 different ways that we can basically alter the expression of genes. The next bit we need to have a look at is how we can predict amino acid sequences. Now, if we've gone through the process of sequencing the genome, then what we have are a sequence of bases. What we also know from our earlier work in Module 2 is that we've got these triplet codons that code for specific amino acids. So if we know the sequence of bases, we can then utilize that information alongside the amino acid triplet codons to then work out what we're likely to see in terms of that sequence of amino acids. So this is then going to give us the probable primary structure of a protein. And what we're able to do there is, just looking at a DNA sequence, we're then able to identify what this unknown primary structure of protein may be. Obviously, to be able to do this successfully, we need to know which parts of that sequence are introns and which are exons. So when we're talking about an intron, this is the non-coding region of the DNA. So these are the bits that we won't express in a protein. Whereas the exons are the coding regions of the DNA which will be expressed. So obviously, if we've just got this sequence of bases and we're just making the assumption that everything is going to be expressed, it's not going to work well for us, because some of those will be the introns. So we need to know which are introns, which are exons, and then using those triplet codons and our DNA sequence, we can predict that primary sequence. The last bit we need to look at is this idea of synthetic biology. Now, synthetic biology is this interdisciplinary science concerned with designing and building useful biological devices and systems. So what we're really looking at here is an overlap of several different areas of science. So we've got the metabolic engineering. So this is obviously using gene networks and building those together. Systems biology, understanding natural gene networks. And something that we may not think about in biology too much, computer science. And that's obviously looking at different data and how we can model and analyze it. And all of those different aspects come together in this field of synthetic biology, because we're going to be utilizing different aspects from those 3 different strands to be able to then come up with useful biological devices and systems for our everyday lives. So what we're really doing in this synthetic biology process is redesigning an organism so that they're going to produce a substance or do something that's useful to us as humans. We're kind of playing around with an organism to make it better for us, is the reality. And there's some great examples of this, and some of these you will have heard about, even going back to GCSE. So one of the ones you probably heard about at GCSE was golden rice. So this is looking at taking one of those staple foods from around the world of rice, but modifying it to solve a problem for us. And the golden rice was all about solving the issue of the lack of vitamin A in people's diets in certain countries, leading to increased rates of blindness, particularly in children. So what do? We modified that rice. We found a gene that was going to be useful in producing the beta-carotene, which our body will then convert into the vitamin A, which allows us to then solve our problem. So basically, we took a gene from a daffodil and we were able to insert it into rice, making this golden rice so that when people consume that, they are getting that supplement in their basic diet and avoiding those high rates of childhood blindness. Another example is using microorganisms for bioremediation. Now, bioremediation is this whole idea of using living organisms to basically clean something up. So thinking about pollutants from air, soil or water, we can use microorganisms for this. there are some microorganisms that love to feast on hydrocarbons. So there's the potential for being able to use microorganisms for clearing up oil spills, for example. Because if we're able to use those microorganisms that feed on those hydrocarbon chains, crude oil is made of a whole array of different hydrocarbons. Is there a way that we can then clean oil spills by basically seeding with microorganisms rather than all of the strange array of things we currently do? And our last one on there, I've given you as an example, not all examples clearly, is that we can use yeast to produce rose oil. And obviously we can grow real roses and then take the oil from them to use in the perfume industry. But in order to get enough oil for this, we need an awful lot of space to grow a lot of rose bushes. If, however, we're able to use yeast to produce the rose oil, yeast takes up an awful lot less space, which means we've then got more land for other purposes, like producing food to meet the needs of an ever-growing human population. The other aspect of our synthetic biology is having a look at information storage. Now, this might be a little bit of a strange idea to think that we can store information within something to do with a living thing, but we can. If you think about your DNA and just how much information it is storing, basically what we're saying is we can store digital information onto a single strand of synthetic DNA. And in terms of the amount of information we're talking here, a single gram of DNA is able to hold the same amount of information as more than 1 million CDs. So just think about the difference in space alone that is going to save us as far as storing information goes. And the reality is we are in a civilization where information is everything, and therefore we are storing huge amounts of it. If you just think about how much information you have stored probably on just your mobile phone, all your photos, videos, all of the different documents you may have, etc., that's going to be a massive amount of information. If we were to put it onto CDs and the like, it's going to take up a huge amount of space. So why not make this synthetic DNA? And the way that we do it is that each letter actually has this 8-digit binary code. So what we're going to be able to do is translate this into a block of 5 letters using A, T, C and G. So if we have a look at a bit of Shakespeare for a bit of overlap with our English there, so we've got Sonnet 18, thou art more lovely and more temperate. We're not going to do the whole thing because that would be a lot of letters on here. If we just have a look at the word thou. So we've got 4 letters there, and each of those letters is going to be divided up into a sequence of 5 bases. So the T is T-A-G-A-T, and obviously we can carry along, just carrying on with our little blocks of 5. So that gives us a way that we can store this information just by using those 4 bases in our DNA. The next useful application of our synthetic biology is in the production of medicines. So we can actually modify the E. coli and yeast cells to produce key medications. And a good example is the anti-malarial drug artemisinin, and this is then going to originally be extracted from a plant at the right point of its entire life cycle, problematic. Now we can just take the actual gene from the plant, insert that into E. coli or yeast through genetic engineering, and they will then produce the drug for us. We've removed that complexity of having to have a plant that's growing. We've removed the complexity of having to have it at the right point of its life cycle to remove the drug. And we've now just got a vat of these bacteria or yeast cells which are going to produce the drug which we can extract easily. Third application is in novel proteins. And basically this is having a look at designing proteins that are very similar to ones that already exist, but don't have certain downfalls. An example we've got here is producing something that's very similar to hemoglobin, but it won't bind carbon monoxide. We can also use synthetic biology in biosensors. So what we're able to do is basically use these to detect certain things. Because if we modify bacteria to show bioluminescence, so basically glowing, when certain things are present, we can then use them to detect certain pollutants being present in a sample. And then we come on to the nanotechnology, which is basically having a look at these little materials that we can now produce that are incredibly tiny materials made from very small particles, hence nano obviously. And a good example here is the amyloid fibers which we use in biofilms, and these are then going to be used in certain things like adhesions. Obviously there's loads of great potential applications for this field of synthetic biology, but it's not all plain sailing. We're opening up an awful lot of potential ethical considerations and biosecurity issues. So what we need are an awful lot of regulations to be in place and advisory panels to actually weigh up the risks that are possible with whatever people are proposing and come up with suggestions on how to basically mitigate those risks. It's not just a free-for-all in the world where you think of something you can do and you just go and do it. We've got to be considering the potential implications for biosecurity. So if you do this modification on an organism, are there likely to be issues? Is it going to create unintended consequences? Hopefully not. That's what the advisory panel is going to do. They're going to weigh up those risks, those benefits. They're going to come up with some things that you are going to have to do to minimise the potential risks there. And there's an awful lot of regulations in place, because when we're talking genetic engineering, that's been around for, 40 odd years now. So we've already got experience of this, and therefore there's a lot of regulations that already exist. But any time that we come up with a new idea, we obviously have to make sure that we are considering the ethical implications and any biosecurity implications. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel for the latest updates, and head on over to my website where you can find a range of other resources with the hope of helping you in your A-level biology studies.